Alright, in this video I wanted to give an overview of several major concepts that we're covering in this section of our course. And luckily, in the field of biology, we already have a theory, an idea, that we call the central dogma of molecular biology that organizes all of these concepts together. So by discussing this thing, the central dogma of molecular biology, we can get an overview of several major processes that take place inside the cell. So let's begin by talking about this idea. So this central dogma of molecular biology, what are we talking about when we use this descriptor? So a dogma is a belief or set of beliefs that is accepted by members of a group, so here biologists, without being questioned or doubted. So a dogma is just a way of saying, here's a set of ideas that we have that describe something, and we strongly believe in this set of ideas, right? So the underlying scientific evidence for all these processes that we're going to talk about is really strong, so we don't doubt it. And so that's why we call this the central dogma of molecular biology. So this overall dogma describes how biological molecules store and use information. All right, so when we talk about this central dogma of molecular biology, we're thinking about some major molecules. We'll identify what they are. And then we're going to think about some processes that allow us to use those molecules to store and use, execute the information in those molecules. All right? So it's probably easier to demonstrate this central dogma by showing it visually rather than trying to describe it with words. So let's build a diagram of the central dogma and what's going on. And what we're going to start with are the molecules, the molecular biology part. So what molecules are involved? We're going to look at three major molecules. All right, so the first is DNA. We've talked about DNA in the past. We know that DNA is used to store information inside of cells. So I'm going to start my diagram with a DNA molecule. There it is with its double helix structure. All right, the second molecule we're going to talk about is RNA. RNA transports information. All right, so DNA stores information, RNA moves the information around. And so I'm going to add an RNA molecule to my diagram here. I'm going to put it below the DNA. All right, so the third molecule we're going to add to our diagram of the central dogma is protein. Proteins perform all the important functions for cells. Right? So cells, they absolutely require protein functions to make things happen. So we're going to add proteins to our diagram. We'll add it at the very bottom. So here's a protein. Each ball represents an amino acid stuck together in a string, and we've got a protein. All right? So our central dogma relies on these three molecules, DNA, RNA, and protein. And now we're going to think about how the functions of these molecules relate to one another. So now we're going to add in three steps or three processes that link these molecules to one another. All right, so we've started our diagram with the molecules themselves, and now we're going to add in some processes. The first process we're going to add in is DNA replication. So in DNA replication, we can take one of our DNA molecules. So we have this DNA molecule here. And using DNA replication, we can create an identical copy of this DNA molecule. So as a shorthand for DNA replication, I said, well, DNA, and then I have an arrow meaning to make, DNA makes more DNA during DNA replication. So let's add that to my diagram right over here. I'm going to put this arrow that feeds back on itself. So we start with DNA, we're going to end with DNA, and the process that makes the extra DNA is DNA replication. All right. The second process we're going to add in is transcription. Transcription is a process by which we can read DNA and then go through this arrow, and we're going to make RNA. So DNA going to RNA is transcription. So we're going to link our DNA molecule and our RNA molecule here with transcription. So transcription reads the DNA and makes the RNA. All right, the last process that we'll add to our diagram links RNA and protein together. So that process is called translation. Translation reads the information in RNA and uses that information to manufacture or produce a protein. So I'm going to add translation right here to link our RNA and our protein molecules together. Translation reads the RNA, produces the protein. So again, we've got three molecules: DNA, RNA, and protein. We can make DNA by reading DNA and then doing DNA replication to produce more DNA. We make RNA by doing transcription, reading the DNA, making the RNA. And then we make protein through translation, reading the RNA, making the protein. So there's one more piece of information I want to add about each of these steps, and that's what enzymes are involved to make them happen. So each of these steps requires an important enzyme to ensure that we can complete that step. Some of the steps require more than one enzyme, but for right now we're just going to think about one major enzyme that makes each step happen. So for DNA replication, that enzyme is called DNA polymerase. So it's here on our diagram, and I've listed it out separately. DNA polymerase is an enzyme that builds DNA polymers, or big chains of DNA. And the name ends in ase. Any name ending in ase indicates it's an enzyme. So DNA polymerase is an enzyme that builds DNA polymers. All right, so that is the enzyme for DNA replication. For transcription, we're going to use an enzyme as well. This one builds RNA polymers, long chains of RNA. So we call that enzyme RNA polymerase. Again, ACE signifies it's an enzyme, and its job is to build RNA polymers. So now we need an enzyme that's going to help us perform translation. And so translation's performed by the ribosome. Ribosome doesn't follow that naming convention I just talked about. I sometimes wish we called the ribosome protein polymerase. It's hidden behind my head here, but I wonder if that's not a more descriptive name. But that's what the ribosome is doing. The ribosome is making a protein, connecting together a bunch of amino acids, making a protein polymer by reading the RNA. So it's important that we remember which enzyme makes each step happen. Okay, so at this point we have our completed diagram of the central dogma. Again, DNA, RNA, protein, and we can make each molecule through the process of DNA replication, transcription, and translation. So let's think about each of those processes with a tiny bit more depth. So we'll start with DNA replication. I'll make another video about DNA replication, and we'll talk about the real important details there. But just as a brief overview, the point of DNA replication is that we replicate DNA to create two identical copies. So here's an image that demonstrates what we're talking about. We start with our one DNA molecule. We're going to separate it apart and use half of it, the red half, as the template to make a matching corresponding second half. We're going to do that on the other one as well. And at the end of DNA replication, we've got two identical DNA molecules. We're going to see when we look at this in more detail that this process is called, more specifically, semi-conservative DNA replication. We'll talk about why in another video. And again, this process requires the enzyme DNA polymerase. All right, so that's DNA replication. Let's focus on transcription and think about a couple important details there. So that, again, the goal of transcription is to copy the DNA for one gene, we'll talk more about that later, into RNA. So we're starting with DNA and we're making RNA. So here's a picture to represent that. In the background here, we've got a chromosome, and then the strands of DNA are strung out here. And we've opened up just a small part of the DNA, and we're copying one piece of the DNA, one gene, into this RNA molecule. And that's transcription. Read the DNA, make this RNA copy of the DNA information. We're only copying one little piece, not the entire chromosome all at once. So if you look up the word transcribe in the dictionary, it says transcribing is to make an exact copy of, and then it says a document or some text or something like that. And I want to point out that this word, transcription, the name we use for this process, if we understand what the word transcribe means, this process makes a lot of sense. So we've opened up the DNA and we're reading the letters that are in the DNA, and we're making a complementary copy in the RNA. So letter for letter, we're copying the DNA into RNA. We're making a transcript, an exact copy. All right, and then the last detail I want to point out is that, again, this process requires an enzyme. That enzyme is RNA polymerase. RNA polymerase builds that new RNA molecule. All right, so we'll move on to translation. Here, we're going to read the information in that RNA we just made and build a corresponding protein. So RNA is the instructions about how to make a specific protein. Here's a diagram showing the RNA and then the ribosome structure and another molecule called a tRNA that are all working together to put together these amino acids, or the growing protein chain. So again, in translation, we read the RNA, we build a protein up here. So if we describe it, we're converting RNA information, which is nucleotides, letters like A, U, G, and C. We're converting that information into a protein structure, which is built out of amino acids. What I want to point out here is that RNAs are made out of nucleotides, proteins are made out of amino acids. Those are like two different languages. So again, the name translation makes a lot of sense. We're translating RNA information into amino acid information. And so that's why we gave this process the name translation. So again, the last detail here, this process requires the ribosome, which is a large enzyme complex which makes translation happen. Okay, so that's an overview of all the processes. I just want to point out one last important detail about the entire central dogma before we end, and that is location, location, location. So let's think about the locations where each of these processes takes place. All right, so our DNA is in our nucleus. So if we want to do DNA replication must take place in the nucleus where the DNA is. That makes perfect sense, right? If we want to do transcription has to take place in the nucleus where the DNA is. And during transcription, we're going to make that RNA copy of a gene. And this is where the important function of RNA comes into play. RNA transports information. RNA moves the information from the nucleus, where it's stored in the DNA, out into the cytoplasm, where the ribosomes are, so that we can manufacture proteins. So transcription happens inside the nucleus, where the DNA is. But translation happens out here in the cytoplasm, where the ribosomes are. So based on the location of a couple important molecules, we know where all these steps happen. DNA is in the nucleus, so DNA replication and transcription have to occur in the nucleus. Ribosomes are found in the cytoplasm, maybe free-floating or attached to the rough ER. So translation has to happen in the cytoplasm. All right, so that was the last point I wanted to make. As usual, I hope you'll ask any questions if you have them, and be on the lookout for additional videos describing each of these processes more in depth. I'll talk to you all again soon.